And it's that realization that actually as humans, there are some things that we probably shouldn't be doing. But the question that we asked the platform, we said, have a look in this area of design space of a few 100,000 molecules and try and find in that space a molecule that is able to recognize the disease cell, recognize a healthy cell, and just kill the disease cell. And we ran 4 cycles of our process until we ended up with a molecule that had a completely non-intuitive design. It is able to exquisitely tell the difference between the healthy and the disease cell. So we can walk these narrow ridges through these complex design spaces and find our ways to molecules that not only are exquisitely performing, but actually will make good drugs too.
So what was step 1 on that journey?
Dr. James Field is CEO and founder of LabGenius and world-leading expert in AI-driven antibody discovery.
And now he's here to reinvent how new medicine is discovered
and unlock more effective treatments for patients.
More than anything else, look, the reason that I do this and I get up every morning is because it is an incredible privilege to be working on a problem where you know that half the people in the UK are going to be affected by cancer at some point in their lives. Every single person in the organisation wakes up in the morning to think we're going to change the way in which scientific discovery is happening, but we're also working towards that future where we can get these molecules into patients and hopefully have a really positive impact.
Back onto your lead program, there are a number of T-cell engagers on the market. What makes you think that yours will be different?
I mean, the T-cell engager story is absolutely fascinating. What we've done that's a little bit different here is that across multiple different targets, and we've published on this now, we can show complete on-off killing selectivity. So using this approach, we have the potential to have a molecule that is not only efficacious, but potentially has a broader therapeutic window, which could.
Did you know that 97% of our listeners are not subscribed to the Leading Beyond the Lab podcast channel? Be sure to hit subscribe to make sure that new episodes with the guests and companies that we're talking to land straight into your feed. Thank you. Hello and welcome to Leading Beyond the Lab. Today I'm joined by James Field, founder of LabGenius, a company using machine learning and automated experimentation to rethink how we discover biologic medicines. Their EVA platform explores design spaces far beyond human intuition, and now they're producing real assets, including LGTX-101, a Nectin-4 CD3 T-cell engager, which recently presented some very strong data at ESMO IO. They've also very recently signed a second multi-program deal with Sanofi, which is an enormous vote of confidence in the large pharma world. So James, great to have you here on the podcast today.
It's great to be here, Lawrence.
So drug development has worked, quote unquote, well enough for decades, the big quote unquote there. And what is it that you found and in your research and in your adventures that made you want to address it in the form of a business like LabGenius?
Well, I would question whether it has worked well for decades.
I thought you'd say that.
Yeah. I mean, this goes back to when I was doing my PhD. I'm a protein engineer by training. Did that at a PhD at Imperial. And one of the things that I immediately realized when I started going deep on protein engineering is that humans are actually really, really bad when it comes to designing new molecules. And that's kind of not surprising, really, when you think about it. We've evolved as humans not to engineer molecules at the nanoscale, but more like how to interact with stuff at the macro scale. And so it became really clear that we needed a different way to approach molecular design. And this journey over the last decade really has been about exploring how can we fundamentally change the way in which we engineer new molecules to create novel therapeutics that you could never have found using conventional methods.
Yeah, and so what was step 1 on that journey?
Yeah, I mean, well, step 1 was actually that first reflection of the fact that humans aren't really good at this stuff, and actually maybe more personally that I wasn't very good at this.
Yeah.
Because I think there's this kind of cognitive bias where the first thing that you do is you look around at your peers and you rank yourself relative to how everybody else is doing. And that's the ceiling upon which you define quality being. Now, I think the realization for me was, number one, I was terrible at protein engineering as a human. I could have invested my career maybe working on 1 or 2 proteins, and maybe after, say, 30, 40 years, built up some intuition around how a specific molecule may work.
The traditional pathway.
Or I said more, it's the case that we're all terrible at this. There's got to be a better way of doing it. And it's that realization that actually, as humans, there are some things that we probably shouldn't be doing. But, when you look around in nature, there is this incredible complexity of biological systems in the world around us. And that's this blind process of evolution that's yielded that. And so really it was also taking inspiration from the fact that it is possible for these non-human systems to create incredible complexity that led me to the really firm conclusion that we had to find a new way to engineer these molecules.
Yeah, it's really interesting because it almost sounds like the spirit of real-world application was derived from not wanting to go deeper and deeper into the protein side of things, and then looking at how that's actually going to impact and drive things like cost collapse or drive quality. What's the primary driver there? Is it a bit of both, or is it?
I mean, I would say it's entirely quality, and actually there's a reason for that, which is when you look at a new technology or a new way of doing things, on balance, it's probably going to actually be less efficient than the conventional methodology. And maybe I'll just give you a really concrete example there. If you look at electric vehicles, okay, now they're performing really comparably to traditional, petrochemical-based vehicles. But historically, they had to find these niche applications where, you would never have taken a petrochemical vehicle, like the moon lander, for example.
Yeah.
You're not going to take you don't install a petrol pump on the moon, and so it was looking at those kind of niche applications. And I think when you have the confluence of new technologies driving innovation, it's around looking for what are the applications that you can't solve using any other methods, and that's a place to focus first. You focus on those areas first, and then after that, it becomes a de facto way of doing things. And I think we're seeing the same technological shift happening across the whole of the world of drug discovery.
And what keeps you going in a long-term cycle or platform business environment?
Yeah, I mean, that's a real challenge, right? In the sense that as any operating environment becomes more competitive, your time horizon typically shrinks down to what is that next milestone that you've got to hit. And, a lot of biotechs, if not all, are financed in this kind of 18 to 24 month venture capital cycles where you've got very near-term milestones that drive that value inflection. But the technological arc of innovation may go beyond that. And so I think the only way to kind of square that conundrum is to bring together not only a team, but a board and an investor base that really buys into that long-term vision. And that's what I think we've been pretty effective at doing at LabGenius. We've got incredibly sophisticated investors, partnered with strategic investors as well. Like our last round was led by Merck's venture arm, M Ventures. The way they look at this stuff, they have this incredibly long-term time horizon, and they're looking at this not only through those near-term value inflection points, which are of course important, but what's going to change the field over the next 5, 10, 15 years.
Yeah, I noticed that actually quite quickly from your investment landscape and syndicate. You've got a real range of different investors. Could you talk about that a little bit?
So the architecture of our investor base actually really nicely reflects the company's origin and its growth. So this is a company that I started when I was doing my PhD at Imperial. And the first question that we really wanted to answer is it even possible to bring together a suite of disparate technologies, gene sequencing, synthesis, high throughput experimentation, automation, etc., together with synthetic biology to accelerate protein engineering? That's a very specific question. And the original investor base that we found that was willing to underwrite that risk was not your traditional biotech investor. These were sophisticated investors, but those who had been really experienced about building deep tech companies, either here in the UK through investors like Backed and Kindred, or in the US, investors like Lux and Obvious, etc. And we brought together that investor base who supported the business through as we built that first iteration of our discovery engine. And then as we gained confidence as a business around the fact that it was actually possible to bring these discrete elements together to find new molecules, we then said to ourselves, now is the time to start investing behind our own pipeline of molecules. And at that point, the risk that you're asking investors to underwrite becomes quite different. You're really asking investors to not only believe in the underlying technology platform, but also to believe that the capital allocation strategy behind these, really discrete molecules is actually going to be something worthwhile and valuable. And that's where we've started bringing in very sophisticated life sciences investors.
And so, complementing that, so with the business structure, how much of it is split between working with, for example, as a service provider and innovating existing trials with partnering with companies, be it small or large, and how much of it is focused on your own or building your own pipeline?
Yeah. In the near term, the primary value driver is investing behind our own internal pipeline of molecules. The lead of that is, or I should say, with respect to our own internal pipeline, this is all focused around oncology, specifically solid tumors, where our lead molecule is a Nectin for CD3 T-cell engager that we'll take into the clinic next year. The beautiful thing about partnering with others is it does a couple of things for the business. Not only is it a nice supply of non-dilutive capital, but it also enables us to expand the scope of the platform into other areas. Fundamentally, what our platform allows us to do is optimize molecules across multiple different properties in parallel. And so that whilst that's useful for us in terms of the work we're doing in oncology, anyone developing novel therapeutics can use that in their application areas. And just by way of example, at the end of last year, we announced another deal with Sanofi, where they're actually, yeah, thanks so much. And what's amazing about that partnership is that they're using our technology in a completely different therapeutic area. So whilst, as I mentioned, our internal pipeline is focused on oncology, for the work that we're doing with Sanofi, that's in the area of inflammation. But the underlying approach that we take across those different areas is exactly the same. So we're running these very high throughput assays, combining that with the machine learning and optimizing novel protein therapeutics across these disparate properties of interest.
And does that all feed into your EVA platform?
Exactly, yeah. So the way this platform works is the scientists sit down together. We can get into the details of the mechanics here, but the scientists sit down together and they say, this is the area of biological design space we want to explore. That is the extent of the scientific input. After that, the whole process runs in this autonomous fashion where the designs are all from that design space are picked out algorithmically, the molecules are made, they're experimentally characterized, and the data is then used to then refine those predictive models. So it's this closed loop lab setup that allows us to very efficiently traverse this design space and actually end up in areas of that design space that you never would have gone using conventional methods. So,
have you got an example?
Yeah. I mean, I'll talk about our lead program actually, because that's the most mature and it's kind of crazy. So, the challenge that we wanted to go out for there is we wanted to go for a target that is expressed on both healthy and disease cells, but create a molecule that had the ability to compute the difference between what is a healthy cell and what is a disease cell. Now, the reason that this is important is because if you look at the way in which our kind of natural biology works, antibodies are made in the body and they work at the molecular level in the sense that their job is to bind to specific targets that they can recognize at the molecular level. The issue is that often the unit of disease is not at the molecule level, it's at the cellular level. And that disconnect between how your natural biology works and the nature of the disease creates a problem. And that problem is the fact that healthy cells and disease cells to your body look indistinguishable a lot of the time.
Okay.
So what we were trying to do here is to create a molecule that could take multiple different inputs and effectively look at the surface of a disease cell, look at the surface of a healthy cell, tell the difference between them, and then only selectively kill the disease cell. So there's this whole range of what's called these dirty targets. These are targets that are expressed on healthy and disease cells. That it's been.
So why are they called dirty?
They're called dirty. Well, a clean target is expressed exclusively on, say, a disease cell.
So easy for human intuition to identify.
Yeah, or easy to create an antibody against, because I just need a binder against it, and I know that if I bind tightly to that and couple on a killing mechanism, I could just kill all those cancer cells. The problem is, if your cancer cell doesn't have any of those markers, then you're limited to going for this other class of targets that are expressed on healthy and disease cells. And when you're going after those types of targets, if you just have a strong binder, you're going to kill your healthy cells, you're going to kill your disease cells. And that, for the patient, is the thing that ends up with all these horrible side effects. So the question we asked ourselves is, by using a platform like this, could we create a molecule that could distinguish between those two cell types? And so in this example, we went after the Nectin target. Nectin is a well-known tumor-associated antigen. It's expressed across a range of different solid tumors, but it's also expressed quite extensively on your skin. And the question that we asked the platform, we said, have a look in this area of design space of a few 100,000 molecules and try and find in that space a molecule that is able to recognize the disease cell, recognize a healthy cell, and just kill the disease cell. And we ran 4 cycles of our process, and with every cycle, we kind of navigated through deeper through this design space till we ended up with a molecule that had a completely non-intuitive design. It's got this really unique, and you'll have to chuck up a picture of this molecule, but it's got this unique 3+1 architecture. So it's got 3 nectin binding units, 1 unit that binds to the immune cell, and it is able to exquisitely tell the difference between the healthy and the disease cell. Now, if you ask any scientists in our team, or scientists I've spoken to outside the organization, they look at this molecule and they say, what on earth is that? I've never seen anything like it. I wouldn't have made that molecule. And the reason most people wouldn't have made a molecule like that is because as a molecule becomes more complex in terms of its molecular design, it becomes a real pig to manufacture.
Yeah.
And so humans have actually been really conservative in the way that we explore this design space historically. But the key part of our platform is we can also co-optimize molecules, not only for function, but also how easy is it to actually make them? How stable are they? How well do they get expressed, etc. So we can walk these kind of narrow ridges through these complex design spaces and find our ways to molecules that not only are exquisitely performing, but actually will make good drugs too.
So do you think that, just thinking out loud, do you think that companies like yours, LabGenius and their unique technology, will apply to the quote unquote dirty cells or dirty targets, so that they can, because it's necessary to cure those, whereas with traditional conventional means of drug development, they'll still be able to target the small antibody type targets? Or do you think there's going to be a world where tech is involved in everything?
I think what we're seeing is at every stage in the drug discovery process, tech is just becoming an intrinsic way in which you run that part of the process.
Yeah.
So I think what we're seeing, and this is happening both within the context of large pharma companies, but also in biotech companies, is that the whole way in which drug discovery is done, right from target discovery to molecular discovery, all the way through clinical trial design, and after that, everything is becoming tech-enabled. And so this is an incredibly exciting time to think, what are the technologies and tools that could massively improve my process? Or is there a new business that I want to start that could take advantage of this? So it's a really exciting time for the field.
It's the best way to start a business, to solve someone's problem, right?
Yeah.
I'm intrigued by that, though, because if I think about search work with classical biotechs, if you like, I can imagine it's very difficult to weave in tech-enabled AI drug development when you consider that they've already got their conventional processes outlined. So to do that natively from the get-go must have significant advantages. But do you lack other things when it comes to the kind of the drug development know-how and things like that people just get through years of experience? Where does that inflection point tee up?
Yeah, it's a great question, and we've kind of had to get to that point iteratively where we have the right balance and the right blend.
Was that when you started your own programs? When you
I
felt that you had that?
I think actually it was. You need to have a certain weight of expertise in the business, and that's basically a critical mass of real expert drug hunters who can work hand in hand with folks who deeply understand the potential, but also the limitations of the technology. And so when we were first starting the business, capital is constrained, the number of people you've got is quite constrained. So you're looking for those people who have maybe like incredible breadth, but maybe not the depth that you then need later on. There's also cultural challenges as well. So when we first
I'm interested by this.
Yeah.
Yeah.
People become a scientist because they want to provide high quality intellectual input. The nature in which that input has to be framed in the context of a drug discovery engine like the one we've built is different. So within a traditional organization, scientists are asked when they're trying to engineer these complex molecules to come up with a handful of designs. Those designs then get made and tested. The approach that we take is we ask scientists to define the area of design space that they want to explore, which could be a few million at this point of potential designs. Those are actually subtly different tasks. And so when we first started running this process where all the designs are generated algorithmically after you set your design space, really experienced scientists would look at these designs that the system wanted to test and say, why are we even testing these? Like, I know this molecule is never going to work. And you know what? They were right.
I was about to
A
say,
lot of the vast majority of those molecules are not going to work. But that's exactly the point.
But some of them do work, right?
They do, but yeah, and what you're trying to feed your model with is that knowledge. It's like teaching a baby. You're trying to show it, here are all of the designs that don't work, here are the designs that work a bit, and here are the designs that work really well. And so when you're doing those first cycles and you're purely exploring a space, actually that's really important. And it only became the case that after we'd run a few of these cycles, and these experienced drug hunters could see that the output of this process were these molecules that had exquisite function but were very non-intuitive, that they stopped saying, you know what, I think we need to test some of these other human design molecules. In the very first few cycles, to stop like a revolt, we had to actually build systems and processes that would allow the scientists to enter their own designs in parallel so that they could compete them in parallel. I think otherwise, like, we wouldn't have been able to actually execute against these projects. And it was only when they saw that this approach, where we're feeding the machine with lots of data, allowing it to make mistakes, allowing it to learn, actually ended up with a better result. That they were then trusted the system enough to let it do its thing.
Yeah, and I can see, like with all language-based AI, ChatGPT type models anyway, in the real world, the more context you give it, the more of the expert drug hunter's information that you can feed into it, the more it becomes better at making those decisions, right?
That's true. I would say a unique challenge that we face, though, is a lot of the time where we're looking at these discrete biological problems.
Yeah.
We're mapping these areas of design space that are very particular to a particular area of biology. So if what we may learn on one target actually may not generalize to another. And this is a really important aspect of biology, because there's a lot of overhype where folks are saying, you know what, we can train a universal model, it's going to generalize to everything and solve all our problems. What we actually find is that to train high quality models that are actually useful from a perspective of generating real drug molecules, actually the key capability there is, can you generate very high quality experimental data quickly? And then actually train like a local model to search a particular area of design space. Yes, there are some things that generalize across projects, but that ability to really generate high quality proprietary data very quickly is a key differentiating factor. And that's actually where, to look at a company that's building in this space and think, do they have a competitive advantage? You have to be confident that they're combining that deep understanding of biology, the high throughput experimentation, and the machine learning together, and they're not just relying on one of those pieces individually.
I was speaking to a KOL in Durham, and he made a really interesting quote, which was most tech bios at the moment have a lowercase b and a high uppercase T, and I think what you've just articulated is the opposite of that. And it does sound like you've addressed both, and that's how which I can imagine takes a lot of iteration and trial and error, but to bring the best out of both sides, which also probably comes down to the cultural elements, because they've got to kind of enjoy each other's ways of working.
You have to bring those two pieces together, and I would say, one of the real pitfalls here is that, if you're a company, as you say, lowercase b, uppercase T in tech bio, like there's a gravitation towards saying, where does the data already exist? Because, maybe you're really tooled up on the computational side. You're looking for open access data sets that you can then train models on. Now, the nuance of biology is, if there's a data set in the public domain, or it's very easy to generate data, it's probably not a problem that you really actually want to use technology to solve anyway, in the sense that biology is incredibly good at running like stuff in multiplex anyway. And I'll give you a very concrete example here. Let's say that you want to find like a new antibody binder against a specific target. There are huge numbers of different experimental methods that allow you to do that at high throughput really quickly, and it's therefore commoditized using existing methods. Yes, you can layer on AI on top of that, but you may just end up with a slower, more expensive process. So I think this is where the really thoughtful application of these technologies, ensuring that you're leveling them at problems that could not be solved using existing methods, becomes really critical. And you don't get that insight unless you have on your team really experienced drug hunters who can say, here are the places where you should innovate, and here are the places where actually the existing methodologies work well enough.
And back onto your lead program, the LGTX-101. So there are a number of T-cell engagers on the market, or there has been. So what makes you think that yours will be different?
Yeah. I mean, the T-cell engager story is absolutely fascinating.
As in within your company or just in general?
Just like in the field in general, right?
Yeah.
There was this incredible optimism around how this new modality was going to transform, the treatment of cancer by redirecting the immune system towards cancer. And they went after a lot of these targets that, as I say, are expressed on both healthy and diseased cells. And what they found is a lot of those molecules ended up being failures on account of the on-target off-tumor toxicity. So either that T-cell engager would bind to and fail to differentiate the difference between healthy and diseased cell, your immune system being brought into and killing not only the diseased cell but also the healthy cell. And that really set the field back by several years. What we've done that's a little bit different here is we've brought in a selectivity enhancement into that molecule. So using this approach of avidity-driven selectivity, which is where we have multiple binders that are able to then effectively differentiate between healthy and diseased cells based on relative receptor density rather than their presence or absence, we have the potential here now to have a molecule that is not only efficacious but potentially has a broader therapeutic window, which could, we hope, enable us to treat these cancers where Nectin-4 is heavily upregulated without seeing the horrible side effects that you get using conventional methods.
Yeah, and so your business model effectively will be to target those types of targets and types of oncology.
Yeah, I mean, for our internal pipeline, we're going after these really challenging targets that you also see on healthy cells. And the beauty of our platform is we're not just doing that in the context of T-cell engagers. We can do that with ADCs and other modalities as well. So within our pipeline, with our internal pipeline, the focus has really been around how do we use our platform to generate that selectivity enhancement that will address the challenge of on-target off-tumor tox. And what we've shown really comprehensively there is that across multiple different targets, and we've published on this now, we can show complete on-off killing selectivity. So we're not traditionally, when people show that their molecules are more selective, what they show is these sort of like a curve shift. So two basically killing curves shifted apart, and that's your therapeutic window. What we've managed to demonstrate is by using this method, we can create molecules with complete on-off killing selectivity. So it's more of a kind of a logic-based approach.
It's really interesting, isn't it? The way, and how does it differentiate between other companies trying to target or do the same sort of thing?
I mean, because this is such a challenging area, selectivity enhancement has been a really active area of research for over a decade now.
But you guys have been definitely one of the most successful.
Well, so, I mean, there are different approaches, and broadly, I would say you could look at conditional activation, and that's things like cleavable masks, pH sensitivity, the approach we're taking, which is avidity driven. These are all great approaches. At a molecular level, there's a place for each of these. What historically has been the challenge has been fine-tuning them so that you could really drive open the therapeutic window. And I don't think that's something that a human can do through rational engineering. And it's through that, I guess, that combination of very thoughtful molecular biology around how we choose which method we're going to use, and then combine that with the machine learning and high throughput experimentation, we've ended up with these quite unusual molecules that seem to have these exquisite properties. Obviously, the next step now is to take these molecules forward into the clinic and actually see, is everything that we're seeing through these kind of mouse models and in vitro studies, will it be recapitulated in the clinic? And that's a big question.
And then when are you targeting to be in the clinic?
Yeah, the goal is to take our lead molecule into the clinic next year. So it's going to be a big, 2027 is going to be a big year. Yeah.
So that's an enormous inflection point.
Yeah. And I think just maybe to build on that, we've got folks who, get out of bed and they're incredibly excited about the fact they're changing the way in which the scientific innovation process is happening. But they're also really excited by the fact that we have this opportunity to actually get these molecules into patients and make a real difference to diseases that frankly affect 1 in 2 people in the UK.
It's incredibly motivating.
Yeah.
You think the amount that's going to be available in the next 10 to 14 years by comparison to what our parents have suffered with and things like that, it's enormous. There's no comparison really.
Yeah, but it's still really frustrating, isn't it? The fact that medicines that we're treated with today, that arc of innovation, maybe those were molecules that were in the lab 20 years ago. You know?
Yeah, that's massive actually, because I think about if you think about exec search, right? When you're placing people in clinical development and what they're working on, and then you might have a relative that's getting chemotherapy or something like that, and you know so much about what's coming out, and even in the nuclear medicine world, by comparison to, as a more direct comparison to chemotherapy, that it is frustrating, and you can see how having access to people is super. It kind of gives you an edge, but equally that should be standardised across the sector. So how do you get in front of that? It's interesting, isn't it?
Yeah, I would say that, a big challenge for the field is, we've talked about the use of technology in terms of doing things that you can't do using conventional methods. Obviously, the other application space for this technology is compressing timelines, and I think
that's where the cost cutting would come in, though, right? Because I was thinking that when you said it was exclusively quality earlier.
Well, I mean, the cost in terms of the cost saving, the biggest reason that this game is so expensive is the late stage clinical failures.
Yeah.
So, if you could be confident that the molecules that you're progressing have a higher probability of success, that is your best way to ultimately save your investment.
And you're going to take your EVA platform all the way through to phase 3, is that right?
Look, for each of these molecules, they'll have their own kind of life history, as it were, depending on how they're positioned, the indication areas that we go for. But certainly, when I think about this business and I think about the potential of the platform, what I'm obsessed by is how do we continually drive this business so we can continue to invest in the platform whilst progressing the pipeline of molecules, and I think that is the challenge for any platform-based biotech. So
tell me more about the Sanofi partnership, a multi-program deal, because that's obviously massive, and the fact that they came back for more means you've done a good job, right?
Yeah. So I mean, the origin of this is actually quite interesting. It starts from the fact that we are interested in building these complex biological molecules, and we use a range of different molecular parts to do that. But one of the most useful molecular parts that we use is something called the nanobody, or the VHH. This is the llamas or the camelid family of animals' answer to the antibody. They're these kind of tiny modular binding units. And very early on in the company's history, we decided, look, these look like very promising binding units to use as a basis of these complex multispecifics. Now, Sanofi did something incredibly smart several years back, and that was the acquisition of Ablinks. That Ablinks was the company that really pioneered the discovery and the commercialization of the nanobody antibody binding agent, and we're privileged that Edwin Moses, who was who's our chairman, was the CEO of Ablinks. So we wanted to partner with the company that was the world's leader in VHH engineering, and that is Sanofi, as a consequence of their acquisition of Ablinks. And what we've done together is we've taken their really deep understanding of the field and combined that with the approach that we can take, where we run these very high throughput experiments, combine that with machine learning, optimize across multiple properties, and together as a collaboration apply that within the area of inflammation. We did that very successfully in a first partnership, and on the basis of that success, I was really pleased that we were able to initiate this second partnership, which is really an expansion of that. So it enables us to continue to work with the Sanofi team on these very challenging problems in the area of inflammation, and bring the very best that the high throughput experimentation and the machine learning aspects of LabGenius platform can bring to bear against that problem in partnership with them.
Can you share one of the results that kind of sits as a metaphor for why you think they wanted to come back?
Yeah, I mean, from that first program, one of the things that struck me as very interesting was they gave us this very challenging project where we had to optimize across multiple different properties in parallel. And we ran this molecule through our platform, ran several cycles, and we ended up finding a suite of molecules that you never would have found using conventional methods. And in fact, our scientists originally said we should never make and test these because I think there's actually a very strong reason why these won't work, why they shouldn't be made. But actually, we went with the algorithm's recommendation. We made them, and they ended up being incredibly high performing. And I think that gave the team at Sanofi this insight into the potential of this as an approach to accelerate the discovery of new biologics. And it's when you see those really non-intuitive findings that you realize, yeah, this is the way in which we should be doing things.
And have you got many partnerships with smaller biotechs as well? Is it exclusively pharma?
We expect to announce our next partnership within the next few months. So I won't say too much.
I thought you could say it here today, live and direct.
I won't say too much now,
Okay.
but yeah, keep an eye out.
What's the key differences there, do you think, around the approach, I suppose, from LabGenius' perspective?
Yeah, I think what's been interesting to me has been how the platform and the pipeline really go hand in hand. And, we've been really, really stunned by the results, these selectivity enhancements we've been able to imbue other molecules in our pipeline with. And when we go out and we present on those molecules at leading conferences, ESMO IO, and we're presenting another one shortly, the inbound that we get and the interest that we get from potential collaborators has been really
amazing.
astounding. And I think that is how, you have to build a platform business. You have to be able to progress your own pipeline. You have to eat your own dog food, right? Demonstrate that, you're putting your money behind programs that your platform has delivered. And then that gives confidence to partners to think, yeah, if these guys are willing to put their own capital behind their programs, this is something that we should be seriously looking at.
Yeah, it's a really interesting ecosystem, actually, isn't it, when you think about it? And do you tend to deal with, is it the CEOs or CSOs? Who's the person that tends to get the biggest resonance when they read the data or they know about your company?
Yeah, I mean, it depends. From on the partnering side, it's all ultimately driven by the scientific expert within the partner organization. And, there's no amount of sort of CEO hand waving or, tech hype that is going to influence those guys. They've got to sit down, they've got to see the data, they've got to speak to our scientists, and that is the thing that ends up, really convincing them when they see the cold, hard data, well-designed experiments, and these beautiful results around things like selectivity enhancement. That's the kind of stuff that I, at least from my perspective, I see having the most resonance.
Yeah, I am seeing particularly from the VC community, any early stage company, preclinical or discovery focused, if they're not completely embedded with AI in its infancy, that they do struggle. There's definitely a trend that they struggle to secure that seed or Series A, by comparison anyway. It's definitely apparent. So behind all of this is leadership. So one thing that really stands out about my story, which is a huge compliment to you, is that you're a relatively young founder and an academic founder. Now, sometimes there has been a trend where VCs can have frustrations by academic founders because they perceive them to have, they lack kind of commercial mindset and commercial acumen. And so at some point, there's usually like an inflection point. You buck that trend. So I'd love to hear your take on why you think that is.
I would say, and this isn't specific to me or LabGenius, but I would say that it's just a general observation. If a company is so dependent on the CEO that it can't function without them, you've built the wrong company. And I would say one thing that we've been really mindful about at LabGenius, and I think this is a function of the fact that, look, this is my first job, right? But
yeah, it's amazing. It's life's work. It's crazy.
Over the last, several years, I've been very fortunate to surround myself with real seasoned experts across every single functional area of the business. These are people who I learn a tremendous amount from every day. And, maybe it was the case, the fact that because this was my first job, like that's the approach I went into this with, which is, this is an organization, can't rely on my knowledge and experience because I just don't have much of it, right? But what I can do is surround myself with an incredibly strong team that can educate me and guide me, and really I see my job really as getting the best out of that incredibly talented group of leaders, not only at the C level and the VP level, but throughout the organization, the functional team leads, etc. Every single one of our departments has somebody who's absolutely exceptional, and I think when you're a 60-person org like we are, like that's the only way to do things scalably anyway, in the sense that, even if I was the expert in every single one of those disciplines, just the idea that I would have to kind of, lead or microman, it's a non-starter, right? So
yeah, I get that. I suppose it's almost accepting that with humility and taking a macro view around what's needed,
which when you think about it is actually sort of like intrinsic to the way in which we approach science as well, right? Which is we're approaching the scientific discovery process from that position of humility, where we're saying, like, as humans, we can't solve all the problems. What are the systems and processes that will allow us to get to the best answers? And we can take a similar approach to organizational design.
So just to kind of wrap things up then, zooming out for a second, there's obviously been huge noise about AI in the healthcare sector generally. Which parts of it do you think are overstated?
Yeah. This goes back to your observation that a lot of folks are interested in applying AI to problems, but they may not have the right biological context to know where to apply them. And for me, like, that's where I see the danger. I see, companies with massively overinflated valuations raising, large sums of money, and I scratch my head and I think, ultimately in this industry, yes, you can raise a lot of money, you can have a high valuation, but gravity is gravity. And that's not something that you can escape. And so I'd say, if you are operating in this space, make damn sure that you're working on a high value problem that is the right problem to be solving, and then say, what is the right technological approach to solve this? Because it may be AI, but it may actually not be AI.
And on a personal level, finally, as you quite rightly highlighted earlier, this is your life's work. It's your first job. You've done an incredible job of it. So tell me more about what keeps you driving on, because I imagine you've supplemented that knowledge gap that you did have with incredible work effort, to say the least, and being able to bring other people in when needed to learn from, and being able to retain that knowledge is probably the next skill set.
Yeah, I mean, I think that is the one thing that, starting a company when you're doing a PhD, you actually have a few things behind you. Number one, you can take incredible risk. You're very unlikely going to be earning less because you're earning nothing. So, from that perspective, there's no big kind of loss in earnings that you have to contend with. I looked at getting a postdoc, and I would never have been able to even get a mortgage on my house because it's a fixed term contract. So actually, from some perspective, starting a company was actually less risky than what may have been viewed as the safer path. But more than anything else, look, the reason that I do this and I get up every morning is because it is an incredible privilege to be working on a problem where that half the people in the UK are going to be affected by cancer at some point in their lives. If you can make a contribution to tackling, that incredibly important challenge, you almost like have an obligation to. And I think it's not only myself that feels like that, but every single person in the organization wakes up in the morning to think, yep, we're going to change the way in which scientific discovery is happening, but we're also working towards that future where we can get these molecules into patients and hopefully have a really positive impact.
And if there was 2 or 3 people that have been enormous points of reference that have motivated you and kept you going, be it board member, colleague, investor, or someone else within the community, who are those people and why?
Yeah, I would say the person who's most impacted my journey, I would say that there were probably 3 people, and I'll kind of list them in chronological order. So I would say first would be my university profs. They were incredibly supportive of me to even sort of countenance messing around, trying to build a company while completing a PhD. And, if it hadn't been for them, this thing would have never got off the ground. The next person would be our seed investor at Kindred Capital, Leila, who took a bet on me as a PhD student and said, I'm actually going to put a not insignificant amount of capital behind this person to go out and build their vision.
Where did you meet her?
Where did I meet her? I think this is a story in itself, but when I was raising our first round of financing, our seed round, I had no idea how to raise money for a company. So,
How old were you when you're doing that?
Yeah, early 20s. So, got a list of investors, went through them. They all said, no, sorry, you're crazy. And my wife, who was pregnant at the time, said, James, you've got until I give birth to get your round of financing done, or you've got to get a real job. And so that was the time bound exercise. And I met Leila as I was quite near the bottom of VCs at this point. And that had pros and cons. It's had pros in the sense that by that point you actually know what you're talking about because you've answered enough questions to understand what you didn't know when you started the process. Cons, I was running out of time. And I met Leila, and within I think 24 hours of meeting her for the very first time, she had done more diligence on me in the business than anyone had in the past, and she was able to come forward with a term sheet. So we got the round over the line. I actually got it over the line from the hospital car park.
Really?
Yeah.
So I love those stories.
I was like a little bit late, but my wife gave me a pass, and so Leila.
How did you celebrate?
How do we celebrate? Get relief.
Yeah. It's funny when people ask that question. That's the first thing that comes to mind.
Yeah.
I know what you mean.
Yeah. But she has been an unwavering backer of both myself and the business over the intervening years through all the highs and lows. And the last person, and by no means the least, would be our chairman, Edwin Moses. I've talked a little bit before around how this guy's a titan in the industry and everything that he's done for the commercialization of nanobodies, etc. But for me personally, he has impacted my personal growth and development beyond anything else I've ever done or tried. He will he'll give it to me with both barrels, where he thinks I can do better, and he'll tell me where I'm doing well. And I think it's that honesty and candor and intellectual rigor and experience, right? This guy he has done it. He has built these companies before, and to be able to work with someone like that, to learn from someone like that, I mean, that has been the privilege of my career.
Radical honesty.
Yeah.
Thank you so much, James.
Yeah, well, thank you, Lawrence. It's been a pleasure.